Please appreciate this presentation has been prepared with all sincerity and to the best of knowledge of students, teachers, and volunteers. We welcome words of encouragement and also suggestions for improvement. For use in academics or any kind of exams, please verify and correlate with your course books. For becoming an associate and/or sending contributions in different subjects including storytelling, please get in touch with your brief profile at baskenjoy@gmail.com. So hello everyone. So today we'll be looking at lesson number 3 for fundamental unit of life for class 9 biology. So in the previous lectures we had covered the main components of a cell. We had covered the plasma membrane, the cell wall, the nucleus, cytoplasm. So now we will be looking at the various cell organelles which allow the cell to function, which allow the cell to basically live. So the main cell organelle is the mitochondria. So basically anyone can say that for anything to be living, it has to produce energy. It has to have energy so that it can basically perform all of its daily activities. So this is what the first cell organelle, mitochondria, is. It is basically the powerhouse of the cell. It basically powers the whole cell and it produces energy. So we'll look at the structure and functions of the mitochondria. So the mitochondria is a kidney bean shaped cell organelle having two membrane coverings. The outer membrane is porous and the inner membrane is deeply folded. So kidney bean shaped, what I mean by that is basically a kidney bean. That's not too good. It's basically a kidney bean. It looks just like this. The outer membrane is porous, meaning that it has pores on it through which things can pass, and the inner membrane is folded. It's folded like this. This is the inner membrane of the mitochondria. So what's its function? Well, its function is to produce energy. That's its main function and that's the thing which it does. So how does it really make the energy? Well, the porous outer membrane allows glucose, fatty acids, oxygen, and amino acids inside the mitochondria. The inner membrane is filled with enzymes which act as biocatalysts. So what is a biocatalyst? A biocatalyst just basically aids in the reaction. It helps to speed up the reaction. That's what biocatalysts basically are. They speed up the reaction. They allow the different reactants to react. So the glucose, while traveling around in the mitochondria, will start reacting with the oxygen. So then when glucose reacts with oxygen, this process is known as respiration. So glucose, when it reacts with oxygen, you can remember this process is known as respiration. Not the breathing kind of respiration like when you breathe in and breathe out. It's the making of energy. This is also called respiration. So when the glucose reacts with oxygen, high energy is released with carbon dioxide and water as waste products. The mitochondria then uses this energy to change the ADP molecule into an ATP molecule. So I will explain this in the next slide. What is ADP and ADP? So in this next slide I will make a diagram and help you explain what I just said. So this is the mitochondria. It's a kidney bean shaped cell organelle. So we have the inner membrane, which is like this. So the outer membrane is porous, meaning it has pores on it. Okay, it will go throughout the entire outer surface. The inner surface is deeply folded. It has all these folds inside of it, as you can see. So outside the mitochondria, we have glucose, we have fatty acids, and we have amino acids. So now what's going to happen is these three, they will go inside the mitochondria through these pores. They will go inside the mitochondria through all these outer pores. So once they go inside, they will be in a matrix. So it's called as a matrix. So matrix is basically a jelly-like fluid. It's basically a jelly-like fluid. So they will be inside this matrix. Now, what else is present inside this matrix are enzymes. There are enzymes throughout this matrix. They're spelled as enzymes. There are enzymes throughout this matrix. So as we looked in the previous slide, the enzymes are biocatalysts. We just looked at that. They help in starting and speeding up reactions. So what these enzymes will do, so what will happen, so also we have oxygen. We can't forget oxygen. Oxygen also goes inside the mitochondria through the pores. So what happens is, near the enzymes in the matrix, we have glucose and we have oxygen, the two ingredients for respiration. So what will happen, glucose and oxygen will react together. So C6H12O6, this is the chemical formula for glucose plus oxygen will give us CO2, H2O, and energy. So these are waste products. But what we care about is this energy. So what happens? Brief overview. Glucose goes inside, oxygen goes inside. With the help of enzymes, they will react and they will produce energy. Now we also looked at how what it does with the energy. It converts ADP. It uses the energy to create ATP. So ADP is short for adenosine diphosphate, and ATP is adenosine triphosphate. So what the mitochondria does is that it makes sure that the glucose and oxygen react. It uses this energy produced to convert ADP into ATP. So what is ATP? ATP is an energy rich compound. It's energy rich. It can store energy. ADP can't store energy. It can't store energy. That's why it's not useful to the cell. But once you add another phosphate molecule, it goes from di to tri, then it becomes energy rich and it has the capability to store energy. So this is basically what the mitochondria does. It produces energy. It produces energy rich compounds known as ATP. Also, you'll hear this sometime, that why the mitochondria is considered a strange cell organelles. Well, they're considered strange as they have their own DNA and ribosomes. So also, if you have DNA and you have ribosomes, once you combine these two, you can make your own proteins. So this is why they're considered strange, as they have their own DNA and ribosomes, so they can make some of their own proteins as well, the mitochondria. So next up, we have the highway of the cell. So what happens in a real life highway? So if I'm in city A and city B, they're both inside a country. So imagine the country as the cell, city A as being one destination, city B being as the other destination, and the highway helps you connect, helps you go from A to B. So this is what the endoplasmic reticulum really is. It transports things from one area to another. That's what it basically does, the endoplasmic reticulum. So its structure and functions. The endoplasmic reticulum is a large network of membrane-bound tubes and sheets. Thus, one function of the ER is to serve as channels to transport materials between various regions of the cytoplasm. We just looked at it, how it can transport things from one area to another. It's also a cytoplasmic framework, providing a surface for some of the biochemical activities of the cell. Now we'll look at this in the next slide, that how does it provide the surface area for the biochemical activities of a cell. So, but the main function is basically for it provides the framework, or it provides the road for things to go from point A, from point A to point B. It connects all the points on the cell. So there are two types of endoplasmic reticulum. ER is just short for endoplasmic reticulum. So there's the rough endoplasmic reticulum and smooth. So from now on, I'm just going to say this RER, rough endoplasmic reticulum, and SER, smooth endoplasmic reticulum. So these are the two types of endoplasmic reticulum. Firstly, we'll look at the rough ER. So as you can see, this is the nucleus of a cell. This is just the nucleus, and the endoplasmic reticulum is situated by the nucleus. It basically, if this is the nucleus, this is the endoplasmic reticulum. It just covers it. It just covers it in a circle like this. So this endoplasmic reticulum, as you can see, this one has these dots on it. As you can see, this one has all these tiny dots. Now these dots are actually ribosomes. You can see over here, they're covered with ribosomes. So it's called the rough ER because it is actually physically rough, as it has these ribosomes on it. So as you can see, this arrow is pointing straight towards the rough endoplasmic reticulum, which has all these ribosomes attached to its surface. So what's the purpose of ribosomes? Well, they are present in every cell, and they are the sites of protein synthesis. Now, protein synthesis is basically the manufacturing of protein inside the cell. Also, then once the protein is manufactured by the ribosomes, they will get transported through the ER to various places in the cell. Now, you might be asking, what is protein synthesis? Well, first step, we eat food. We eat everything. We eat roti, we eat dal, we eat chawal. So inside all of that, we have carbs, we have proteins, we have fats. But for protein synthesis, we only care about proteins. We will disregard the carbs and fats. So once we eat the food goes to our stomach, and then the stomach, small and large intestine, the digestive system basically breaks down these complex proteins into simpler forms of proteins. So it goes from proteins to polypeptides, this is just the name, to mini peptides, and then finally to amino acids. So amino acids is the basic form of protein. So the digestive system basically breaks down these proteins, and then the amino acids enter our bloodstream. Then they enter our cells, and then they come inside the ribosomes, where they get connected back together into polypeptides and then proteins. So this last step, step 5, is protein synthesis, just joining of various amino acids, just connecting them to form a protein. This is what protein synthesis is. Now, how does it happen? Like, how does the cell know how to synthesize the protein? Well, we have the nucleus. As we discussed in earlier chapters, the nucleus has DNA. DNA controls the activities of the cell. It's basically the main guy. He's the main guy who controls everything. He knows what to do. He's the guy in charge, basically. So the first thing you need to know to understand what is protein synthesis, the difference between DNA and RNA. So what's the difference between DNA and RNA? Well, you can just look at their full forms. This is deoxyribo, and then this is just Na is nucleic acid. Well, RNA is ribonucleic acid. So DNA is deoxyribo, this is just ribonucleic acid. So the main difference, however, comes in the structure also. So DNA forms a double helix. It's basically two lines like this. This is DNA. RNA, however, is only a single strand of DNA. RNA is only a single strand of DNA. Okay, so this is DNA. This is RNA. DNA is equally as important as RNA, and RNA is equally as important as DNA. So now, how does protein synthesis actually happen? So what the DNA does is it sends some RNA to ribosomes. The DNA instructs some RNA to go to these ribosomes. Now this RNA is known as messenger RNA. So this DNA sends messenger RNA to ribosomes, and what this messenger RNA does is it tells other RNA, so there are other RNA which carry the amino acid. So you can think of this as, you can think that this person over here is RNA, and it's basically carrying on top of its head an amino acid. Okay, so what this messenger RNA does is it tells these other RNA molecules which are carrying these amino acids, it tells them in which order to line up, it tells them which amino acid we require in the ribosome. So the DNA is the head, it sends out its little associates, okay, and these associates tell the workers which type of amino acids do they want to, which they need to carry and bring to the ribosomes, and then in the ribosomes they just get connected to form proteins. So this is the whole process. So DNA tells messenger RNA to tell the RNA carrying the amino acids in which order to line up, and then which amino acids we require, and in the ribosome they just get connected with bonds, chemical bonds. They get connected to form protein. Now this is a common question that first we have protein, we eat protein, we convert it into amino acids, but then why are we converting it back to protein then? Isn't this just a waste of time? It's just a cycle, it's just a useless cycle. We eat protein, we convert it to amino acids, then we just want it back as protein. So the answer lies in the construction of our human body. So the blood vessels inside our human body are so tiny, they're so tiny that big protein molecules cannot fit. First of all, they cannot fit inside the bloodstream, and for the blood cells to carry, and the blood cells can't even carry such big, just these humongous protein molecules. So that's the first reason why we break the protein into amino acids. And the second reason is that we discussed in earlier classes what the plasma membrane is. The plasma membrane is semi permeable. So it's semi permeable, so basically even the plasma membrane, it also says that, hey, we don't want any protein coming in. It says no to protein. It says, okay, amino acids, you can come in. So this plasma membrane also only lets amino acids come in. So that's the reason why we convert it to amino acids, and then but our body can use protein, so then we convert it back to protein for the body to use. So next up we have the smooth endoplasmic reticulum. So this one was the rough. Now this one is the smooth endoplasmic reticulum. As you can see, it has no dots on it. It's perfectly smooth. That's why its name is smooth endoplasmic reticulum. So like we just said that the rough endoplasmic reticulum makes protein. The ribosomes on it make proteins. What the smooth endoplasmic reticulum does, it makes lipids. It makes lipids, which are basically fat molecules. So we don't have to go much in detail that how does it make these lipids, because it's not that important and it's for higher classes. So now we have SER making lipids. We have RER making protein. And we know that the membrane, the cell membrane, plasma membrane, whatever you call it, it's also made of lipids and proteins. So it's a instinctive question to ask that then how is this membrane constructed? So in reality what happens is SCR gives some of its lipids to the membrane, RER gives some of its protein to the membrane, and this is how the membrane is formed. And this process is known as membrane biogenesis. The construction of the plasma membrane happens with the help of RER and SCR. So now we looked at the endoplasmic reticulum, but now we also have another cell organelle, the Golgi apparatus. So you can compare it to a post office in real life, the Golgi apparatus. So you can think, what do you do in the post office? You just basically give them the letter, or you give them the box, whatever. So let's say you give them the letter. They will pack the letter, arrange the letter, and then send the letter. Now if you're living, let's suppose, in one city, let's say you're living in one neighborhood, so you're living in this town, and you're living here and your neighbor is living here. Well, you don't have to give a letter to him, you can just go on walk and just tell them whatever you need to say. But what if you need to talk to someone over here? He's in a different city. He's in a different city. He's in a different city. Now you can't just walk hundreds of kilometers just to talk to him. What you will do, you'll send a letter. And then what the post office does, it will pack it, arrange it, and send it. So this is what the Golgi apparatus basically does. So the endoplasmic reticulum is within the cell. It's for intercellular communication. It's basically for him to talk, this person to talk to this person. He's within the same neighborhood. But if he wants to talk to a completely different guy who's across the country, he won't just walk there, he will send a letter. So this is what the main function of the Golgi apparatus is. It will basically it will allow for packages to be sent between cells. So this is an image of the Golgi apparatus. So now we'll look at its structure. So the Golgi apparatus is a system of membrane-bound vesicles arranged approximately parallel to each other in stacks called cisterns. Now probably none of you guys understood this, and I get it. So what they're trying to say, these are the membrane-bound vesicles. These over here are the membrane-bound vesicles. So are arranged approximately parallel to each other. So as you can see, this one's like this, then this, then this. They're arranged parallel to each other in stacks called cisterns. So this is basically a cistern, you can say, in a way. They're arranged parallel to each other in stacks called cisterns. So this is basically the structure of the Golgi apparatus. So you can just simply draw it like this. You can just make it like this. So start off big, then smaller, and then just top it off like that. So this can be a drawing of the Golgi apparatus. So its functions, well, I already told you back there that what are its functions. So the materials synthesized near the ER are packaged, dispatched to various sites inside and outside the cell. So its functions include storage, modification, packaging, and also sometimes complex sugars are made from simple sugars inside the Golgi apparatus. You don't have to go in that much detail about how these sugars are made. All you have to realize is that we can have smaller sugar molecules. So basically we have smaller sugar molecules, but then they will basically get connected to each other and they will form a larger sugar molecule. So next up we have lysosomes. So no one really, and even in real life, no one really wants to live in a dirty place. Everyone wants everything clean. So this is the main function of the lysosome. It is the cleanup crew of the cell. So its functions are to help the cell clean by digesting any foreign material, as well as worn out cell organelles. So the structure is basically a circle. a sphere. It's basically a sphere. Oh, sorry. It's a sphere, and inside here, there are enzymes, as you can see. There are enzymes inside the lysosome. So what these enzymes do, they digest any worn out material, any bad things, and they can also they digest food. So this is basically the structure and functions of the lysosome. This is all that basically does. It basically just cleans up the cell. However, they also have a really important role, and this is why they're called suicide bags also. So when there is disturbance in the cellular metabolism of the cell, maybe the cell got hurt or something, then the lysosomes will burst, resulting in the enzymes digesting their own cells. So this is the line why they're called suicide bags, because if something bad happens to the cell, they will digest their own cell. So this is why lysosomes are known as the suicide bags of the cell. Also, it's important to note that lysosomes are made by the Golgi apparatus. Thus, this is another function of the Golgi apparatus, the making of lysosomes is another function of the Golgi apparatus. So you can see that lysosomes are basically a circular structure. Then if you go back to here, you can see there are the circles. So these are the lysosomes made by the Golgi apparatus. So next up, we'll look at the plastids. So this is really important, that plastids are only present in plant cells. So don't just label plastids in an animal cell, because they don't exist in an animal cell. Plastids are only present inside plant cells. So there are two types of plastids. The first one being chromoplasts, second one being leucoplasts. This should be second. Second one being leucoplasts. So chromoplasts are colored plastids. The ones containing the pigment chlorophyll are known as chloroplasts. So this is a tongue twister basically. So we have chromoplasts, and chromoplasts are colored. The chromoplasts containing the pigment chlorophyll are known as chloroplasts. So basically, chloroplasts are inside chromoplasts. There are various other chromoplasts, but we are most familiar with chloroplasts. They contain chlorophyll. Also, chloroplasts don't only contain chlorophyll, which is green in color. They also contain various yellow and orange pigments. That's why you might see some yellow leaves, you might see some orange leaves also. This is because they also contain other pigments, but it's mainly chlorophyll present inside chloroplasts. So we have the colored plastids, chromoplasts, but we also have colorless plastids, which are known as leucoplasts. So these are colorless plastids, and they are basically organelles in which materials are stored. Starch is stored, oils are stored, protein granules are stored. So leucoplast is mainly for storage. Chromoplasts are mainly for photosynthesis. So these were the two types of plastids. And things to note about plastids, and this should also be a 2, my bad. So plastids are very similar to mitochondria. It too has a porous outer membrane. It too has these pores on its outer membrane, and it also has its own DNA and ribosomes like the mitochondria. So we just saw that the mitochondria has its own DNA and ribosomes. Also, the plastids have their own DNA and ribosomes. So even plastids can make own protein as required. So the only difference between between the mitochondria and plastids are basically their internal configuration. As we saw in the mitochondria, It's basically folded like this. It was folded. However, as you can see in this image of the chloroplast, there are various membrane layers like this. There are various membrane layers which are embedded in this material known as stroma. So this is the main difference. Just their internal configuration is the main difference between plastids and mitochondria. The rest is basically the same, and like mitochondria produce energy, similarly plastids also perform, the one with chlorophyll at least, perform photosynthesis. So they produce food, this produces energy. Now don't be thinking that okay, there are chloroplasts inside a plant cell, so this means that they won't have mitochondria, but that's actually wrong. They need the mitochondria for the energy. They need mitochondria for energy. However, like us animals, we eat the food. So the food is already put into the cell. We eat the food. However, plants can't move, they can't eat, so they make their own food with photosynthesis. So this is why they have plastids. So now we'll be looking at the last cell organelle, the vacuole. So though in its comparison for real life are storage containers. So like in our homes we have storage containers. In one you might have cookies, in one you might have cake, one you might have some sort of dal. So basically we have containers in which we store things. So even inside a cell we have vacuoles which act as storage containers. So the difference in the vacuoles in plant and animal cells is, so this is for plant, oh sorry, only this first line is for plant, this first line is for plant only, that vacuoles take up 50 to 90% of the space inside the plant cell. So basically if you take a look at a plant cell, you will see that the vacuole takes up this much space. This is a plant cell, the entire thing is almost taken up by the vacuole only. Okay, also the vacuoles are full of cell sap, provide turgidity and rigidity to the cell. And now these 2 and 3 points are the same for animal as well. So they are full of cell sap, they basically provide strength to the plant or the animal cell, and they also, in plastids, many important substances are stored, such as amino acids, sugar, organic acids, proteins, etc. So basically they act as storage containers for these various things. So as I just said, everything is same. These 2 points are the same for animal cells. However, like in plant cells, we only have one large vacuole. We only have one large vacuole. In an animal cell, we have 2 or 3 vacuoles, but they're really small. So one might think that why do plants have a big vacuole, but animal cells only have small, tiny little vacuoles? Well, the answer lies in their lifestyle. In the lifestyle of plant, sorry, it lies in the lifestyle of plants and animals. So what are plants? They are sedentary. They cannot move. So they have to store. So basically, because they cannot move, all of the energy they're producing, all the food they're producing, it's going to get accumulated inside the cell as they're not using it because they're not moving. This is why they have a large vacuole. However, animals, we move, we we can run, we can go to places, we can move. So basically we're using up more of the energy we produce. So this is why we already have little to store, because we used everything up in our movement. We have little to store, we have little left to store. This is why we have small vacuoles inside animal cells. So now we have vacuoles in unicellular organisms. So as stated in previous lectures, we took a look at the amoeba and how if this is food, it's present near it, what will it do? It will basically encapsulate the food inside itself. Now once it goes inside, so it basically sees the food, it envelops it, and then it captures the food. So inside the cell, this is the food vacuole. These are just tiny little differences for unicellular organism. So basically in unicellular organisms like amoeba, when they eat the food, it forms a food vacuole, and then the enzymes will come inside and they will digest the food inside the food vacuole. So then also they also have a waste vacuoles. It's not written here. They also have waste vacuoles to expel excess water, some waste. So once this food is basically digested, once it's digested, these are all the this is all the waste accumulated. So this waste vacuole will basically just dump out all the waste outside the cell. So they're just tiny little tweaks here and there for various organisms. However, the main thing is they just store things. May it be helpful things or waste, but they just store things. That's the main thing about vacuoles. So I know today was a really long lecture, so I'll do a quick recap. We looked at mitochondria, how they are the powerhouse of the cell, how they make energy. We looked at we also looked at how they have their own DNA and ribosomes. Okay, then we looked at endoplasmic reticulum, how it's the highway of the cell, it transports material. We looked at SER makes lipids, RER makes proteins, how protein is made, protein synthesis. We looked at how some of the lipids of the SER, some of the proteins of the RER convert, help in making of the membrane, which is membrane biogenesis. We looked at the Golgi apparatus, the post office of the cell, and how it also makes lysosomes. We looked at lysosomes, how they clean the cell, and their suicide bags of the cell. And then we also looked at plastids, two types of plastid. We looked at chromoplasts, colored plastids, leucoplasts, not colored, or in a way colorless. And then we looked at the similarities with mitochondria, the similarities of plastids and mitochondria. Then lastly we looked at vacuoles, the differences between vacuoles in plant and animal cells, and the unique tweaks of vacuoles in unicellular organisms. So next time, in the next lecture, we'll be looking at the comparisons between animal and plant cells, their organization. So what this basically means, now we know what is mitochondria, we know what is plastid, vacuole, etc. We know all the cell organelles. So now what we will do in the next class, oh sorry, that's really bad, we'll look at animal cell, we'll look at plant cell, and then we'll basically stuff all these guys inside here. We're gonna stuff them, stuff it with cell organelles, make diagrams for the animal cell, make diagrams for the plant cell, compare and contrast them, their differences, and then lastly we'll look at cell division. Cell division. How do cells reproduce? And then after that, this chapter will be over. So that's it for today, and I'll see you next time.